After learning about DNA replication, now we will go to the expression of DNA in living organisms, that is in the form of the transcription, translation, all these concepts we will be learning in our coming lectures. Let us start with it. First, we go to central dogma of molecular biology. What does it mean? That is how genes express themselves, is the controlling, the main factor behind molecular biology. DNA, which exists, which is the genetic material in most of the living organisms, as we have already seen in our previous lectures. The DNA replicates on its own. In the previous video, I had shown that, I had explained about it. So DNA replicates on its own to produce multiple copies, to produce many copies during the S phase of cell cycle. The DNA, which is present in the nucleus, undergoes transcription. Now, transcription is the process of formation of RNA. Now, process of formation of RNA here means RNA, which is a single-stranded polynucleotide. Single-stranded polynucleotide. Now, it does not mean that DNA breaks down to form the RNA. What happens? The DNA will just pass on its codes. It will send a message that such type of codes are existing within it, and taking them as templates, new RNA is produced. That process is known as transcription. DNA remains intact, no change in the DNA. It simply produces a copy similar to itself, but not DNA. It will produce a RNA. Similar to itself means similar to at least one of the strands. That process is known as transcription. The RNA now, the transcription process, it takes place in nucleus in case of eukaryotes and within the cytoplasm in case of prokaryotes. After the RNA is formed, it is passed on to the cytoplasm. Ultimately, its fate or whatever events are to happen, it will take place in the cytoplasm. So, in case of prokaryotes, the site remains the same. But in case of eukaryotes, the RNA formed in nucleus will be transferred, or it will pass through the nuclear pores out to the cytoplasm within the cell. In the cell, RNA will help in the process of, or rather not help, it will carry out the process of translation. Translation is the production of proteins. Now, proteins are the ultimate expression. Proteins are the ultimate expression of DNA. So, the protein that will be produced will be according to the codes that were, the nitrogenous bases that were present in the DNA. So, via RNA, the codes, the pattern of codes is conveyed to the cytoplasm. In the cytoplasm, amino acids will link up one after another to form a polypeptide, maybe in the primary form, maybe in the secondary form, tertiary or quaternary, according to the requirement of the cell. And that protein will be the expression in our body in different forms. It can be in the form of an enzyme, it can be in the form of a pigment, a hormone, and or like say the muscles. Anything you talk about inside a living organism is all made up of proteins. So these proteins, how are they formed? They are formed with the help of RNA. RNA, how is it formed? It is formed with the help of DNA. So ultimately, if DNA is missing, DNA is not there, then even protein formation is not there. So the flow of information is from DNA to RNA, RNA to protein. This is one-way flow. Never protein can form RNA or RNA can form DNA, except in one or except in some cases when we call it as reverse transcription. Only RNA can form DNA, not protein. The RNA can form DNA. That is seen in case of retroviruses, where an enzyme called as reverse transcriptase is present that can convert RNA to DNA. So this is the central dogma of molecular biology. Then in prokaryotic cell, as I was telling, okay, in prokaryotic cell, the transcription, that is formation of DNA to RNA, then RNA, this messenger RNA. Okay, RNA are different types. We will learn them in the coming lectures. Okay, so this is the type messenger RNA. It will undergo translation to form the polypeptide upon the ribosome. So we all are familiar that ribosomes are the protein producing organelles within the cell. So it is formed within the ribosomes translation, and it is taking place within the cell in the cytoplasm itself. In eukaryotes, what happens? Eukaryotes have a proper nucleus. So the DNA, it will undergo transcription to form the RNA. Now RNA, it needs to be processed in case of eukaryotes. First is formed the precursor of RNA, pre-mRNA we call it, and then that will undergo processing, produce the final copy, which will be shorter compared to the pre-mRNA. Even these things we will be learning in the next classes. So mRNA, it will move out to the cytoplasm. In the cytoplasm, it will attach upon the ribosomes and then produce the polypeptides. So ultimately, messages of which molecule is coming out? It is the DNA. It is passing its message to in the form of proteins. Now coming to transcription. So what is transcription? Transcription is the process of copying genetic information from one strand of DNA into RNA to form the RNA. Here, even in this case, complementarity rule governs the process. That is A, T, G, C. That is the complementary rule. But with one difference, because in RNA we do not find thymine. It is replaced with uracil. So in the process of transcription, the complementary rule will be A U or U A U U A G C C G. That will be the complementarity rule. But it remains the same. Just the difference is T is replaced by U. Here, thymine is replaced by uracil in RNA. One strand of DNA is copied. Not both the strands are copied. Only one strand of DNA is copied. Why both the strands are not copied? Because if suppose both the strands are copied, both the strands form RNA, then what will happen? Again, the RNA will also be having the complementary strands. That is one strand of DNA, the complete strand of molecule of DNA. Suppose it had ATGC, just two bases, two pairs I am talking about, ATGC. So one of the strands, A and G, it forms an RNA with U and C. The other strand also, it produces the other. Okay, I'll show with the help of some okay, so let us say this is the DNA which has the bases A, T and G, C with the help of hydrogen bonds. Just I'm showing two bases here, two pairs. Now let us imagine the case which never happens actually, imagining that both the strands are being copied. So here, suppose the first strand, what is it? It has A and G. A and G, so complementary to A in case of RNA will be U and C. So one strand of RNA formed, fine. Second strand of RNA, if the other one acts as the complementary, then what will happen here? The base will be A and G. Fine. So two RNAs are formed. RNAs have to be single stranded in order that they can express. Now as you can see here, the two new RNAs formed out of the two templates from the same DNA will be again complementary. If the complementary possibilities are there, then they may form a hydrogen bond again. What will happen? The entire exercise will be futile. No RNA will be formed. That means it will become a double-stranded structure, and double-stranded structure cannot behave, cannot function what RNA is supposed to be functioning as. So that is one of the reasons why both the strands are never copied. Secondly, there is one reason that is suggested, and the second reason is that probably why both the strands are not copied, because if both the strands are copied, both of them will undergo translation. Assuming they are not forming a double-stranded structure, both of them will undergo translation, and both of them will produce amino acids. Both of them will produce two types of proteins, which will complicate the entire metabolic activity inside the body. So these are two probable reasons why both the strands are not copied during transcription. The enzyme is responsible is DNA dependent RNA polymerase. Like replication, even here polymerization takes place in 5 to 3 direction. Now, transcription unit. Now, what is transcription unit? Transcription unit is that portion of the DNA that will undergo transcription. The whole of DNA does not undergo transcription at the same time. It many portions may be undergoing transcription simultaneously, but each one is independent of the other. The portion of DNA that is undergoing transcription means is forming RNA at a particular point of time inside the body, inside the cell. Some may be continuously forming, some may be according to requirement of the body. So whatever portion is forming the RNA, that portion we can regard as a gene. That portion can be regarded as a gene. So assuming that this whole strand of DNA, here you can see it is here 5 to 3 and 3 to 5. The two strands of DNA are there. Okay, always you have to keep in mind that these are made up of bases, opposite complementary bases, A, T, G, C or CG. That way it will be made up of. Now what will happen? The transcription unit, the portion that will undergo transcription, is always flanked by two additional parts. One part which is in one part where the polymerase comes and binds, and the other part where the polymerase will get detached from. The part where the polymerase, RNA polymerase, DNA dependent RNA polymerase binds is known as promoter region, and after binding what will happen? It will proceed. Okay, it will break the hydrogen bonds initially, and the whichever is acting as the template, one of them will act as the template. The direction in which it is moving, the template strand will help in formation of the RNA. The other opposite one will be called as coding strand. It moves on till the complete structural portion, we call this a structural gene, has completed transcription. When it reaches the other end where it can no more remain attached, that portion is known as terminator. So transcription unit will include a promoter region, a structural gene and a terminator gene. Formation always takes place in 5 to 3. So the DNA strand with opposite polarity will behave as the template. It will be determined with respect to the position of promoter. Promoter is present here. Structural gene is followed structural gene follows the promoter, so definitely formation will take place. That means the polymerase will move in this direction from left to right in our figure. So in this case, what will happen? If it has to move from left to right, then the blue strand has to act as the template, because it will be with opposite polarities to the direction in which RNA can form, that is 5 to 3. So the blue strand will act as template and the other one will be called as the coding strand. So what is promoter? Binding site for RNA polymerase. Structural unit codes for proteins, and terminator is the point where transcription ends. And formation, like if we have here say that they are made up of the original DNA, it is made up of say A, T, G, C, C, G, we are just imagining here T, A, let us say, so on it goes. These are the complementary strands of the DNA molecule. Now when the polymerase will come and bind here, the polymerase comes and binds, it starts moving. As it moves, what will happen? It will break the hydrogen bonds. It breaks the hydrogen bond and nucleosides from the surrounding. Okay, what will happen? It will come and line up here. What is complementary to A? Complementary to A in case of RNA will be U. So one nucleoside with uracil, next a nucleoside with cytosine, a nucleoside with guanine, one with adenine, they will line up now here. Okay, taking the complementary strand, that is the template. Okay, complementary to the template strand, they will go on lining up. It goes on and on till it reaches the terminator. Of course, simultaneously the polymerase is also moving. It moves, and finally when it reaches the terminator, polymerase moves out. Polymerase moves out, setting the RNA free. So you will get the RNA, it will come out with this basis. So it will come out with this basis, then whatever the other basis, it will come out. And the original DNA will continue to maintain its own basis, will continue to maintain its own structure. So this is about transcription unit. In the next lecture, that will come up, we will talk in details about how transcription actually takes place.